based on the induction of callus. Next one is called it as embryogenesis. What is the meaning of embryogenesis? Simply we say formation of embryo with the help of callus, that is called it as embryogenesis. The callus cells undergoes differentiation and produces somatic embryos known as embryoids. That embryoids that can be able to develop from the callus, whether it is directly or indirectly. The embryoids are subcultured to produce a plantlets. Formation of the embryoid is the basic for the formation of the plantlets that can be able to develop into a plant. Next one is called it as hardening. What is the meaning of hardening? The name itself you know. Hard. The entire process that can be able to occur inside the laboratory condition. But the plantlets slightly or slowly that can be able to enter in a field condition. That process is called as hardening. The plantlets develops in vitro, requires a hardening period, and so are transferred to greenhouse or hardening chamber and then to normal environmental conditions. The gradual exposure of in vitro developed plantlets in humid chamber in diffused light for acclimatization. What is the meaning of acclimatization? Without climatic conditions, that is that condition is differ from laboratory condition, so as to enable them to grow under normal field condition. A simple process of the plantlets that can be able to stepwise manner that plant from laboratory to field condition. That process is called as hardening. These are the steps followed by the formation of the plantlets in a tissue culture technique. Next one is process of plant tissue culture. What are the process? The process that can be able to classified into the culture type that can be able to classified into four major types. First one is Argan culture. Second one is Meristem culture. Third one is Protoplast culture. Fourth one is cell culture. First thing, Argan culture. What is the meaning of organ culture? Yes, the organs, that is embryo, anther, ovaries, roots, shoots, or other organ of a plant that can be able to develop from the culture medium with the help of a tissue, that is called it as a organ culture. Here one of the simple diagram. The leaf is act as a explant, that they can be isolated the cell, that the cell that can be able to inoculated inside the culture medium. Then the inoculated culture medium, they can be able to form the plantlets. That plantlets are separate with one another and it can be able to hardening process. That a simple process that can be able to occur in with the help of any one of the organ. That is called it as organ culture. Next one is called it as Meristem culture. The name itself you know, meristem. We want to select the meristem for the culture. The meristem cultures are free from virus especially. Then only we want to select the meristem culture. Culture of any plant material tissues on a culture medium. We want to select the meristem plant, especially in a older part, that they contain a meristem that can be able to placed in the nutrient medium with the help of paper wick. Then the callus that can be able to induced. Then the plantlets that can be able to develop from the shoot tip, that is nothing but a meristem with a growing tip after removal of enclosing the leaves. Next one is called it as Protoplast culture. Using protoplast, we want to culture. We can obtain a plantlets. There are four major steps for the protoplast culture. First one is isolation of protoplast. Second one is fusion of protoplast. Third one is culture of protoplast. Fourth one is selection of somatic hybrid cells. First one, isolation of protoplast. How we can isolate the protoplast? First thing, we want to select a small bit of plant tissue, especially like leaf tissue are used, because we have to use root, shoot, leaf, flower, fruit, seed, anything part. But here we want to select the leaf tissue. Then the leaf tissue is normally washed with running tap water. After that, it can be able to peeled with the help of knife. Then the leaf tissue, that peeled tissue, that can be immersed in 0.5 percentage macrozyme and 2 percentage Onzuka cellulase enzyme. These are the enzymes that can be able to isolate the protoplast present inside the cell. Then dissolved in 13 percentage s orbital or mannitol at pH 5.4. That pH 5.4 that can be able to maintained. Then incubated overnight at 20 degree Celsius, 2 degree Celsius. Then a gentle teasing of cells protoplast are obtained. The cells we want to isolated. After that, it can be able to obtain the protoplast. The obtained protoplast are transferred to 20 percentage sucrose solution to retain their viability. Sucrose solution to retain their viability. Then after centrifuged, the getting the protoplast, they can be able to centrifuge to get pure protoplast are different forms, debris of cell wall, the supernatant solution as well as debris that can be able to separate with one another. Then the supernatant solution, they contain the pure protoplast. Next, how we can fuse the protoplast with the help of fusogenic agent. The fusogenic agent is nothing but fusogen. It is done through the use of a suitable fusogen. The fusogenic agent is nothing but PEG, polyethylene glycol. Using the polyethylene glycol, the isolated protoplast are fused with one another. The isolated protoplast are included in 25 to 30 percentage concentration of PEG with calcium ions, because we have to protect the protoplast from other damages. Then the protoplast show agglutination, formation of clamp-like structure, and fused with one another. Here the entire process of protoplast culture that can be able to explained later in their slide.